the Camellina genomics and breeding team, genetics team, to give us an update on their work. And before I turn it over to David Marks for the introduction and a few words, this is a program that has been developed in parallel with the Pennycrest program and some of the same players. So it's been a network of activities across Pennycrest and Camellina, and over time, Camellina kind of received less of an emphasis. And I think what David will tell you is that we're now moving into an era where it appears that Camellina is going to get additional support and visibility over the next few years. So the oilseed crop program, it appears, is going to have a two-pronged approach, bringing along Pennycrest and Camellina as well. So I look at the team as coming together, having the capacity, I think, to move Camellina along quite effectively over the next number of years. So just some of my thoughts, and I'll now turn it over to David Marks. That has been, his lab has been providing leadership for this overall program. David.
Yeah, thanks, Don. Yeah, mainly I just want to introduce Matthew to you all. First of all, just to say that, the Forever Green biz is all about creating a Forever Green cover. And this is what Don's expertise has really been key to this, in putting together these diverse teams to work on these problems. And the thing that's really great about this is it's a wonderful opportunity for students involved in this to receive cross-training from a lot of different areas. And I think Matthew's embraced that approach very nicely. Matthew began here and got an MS degree in the ABS program. And even there, he was doing nitrogen studies, enterprise studies to look at issues of the economics of the systems, all while creating mutants in Camellina. And so I think that kind of captures that spirit. He's continued with me to work with both Camellina and Pennycrest, as Don said. And I think both of those things are advancing nicely under Matthew's work. One thing I'll note too, just to kind of add a collaboration that's come into play, is that Matthew's picked up a collaboration with Gary Gardner to study the role of light signaling in the development of these cover crops. And I think that's really nice. So, I mean, the thing I really like about Matthew is from the get-go, when I first met him, he just has a can-do attitude. He takes his own approach and tackles important problems. And I think, as you'll see today, he's made a lot of progress. And with that, Matthew, I'll turn it over to you.
Great, thank you, David, appreciate it. So I'll go ahead and share my screen. And start the slideshow here. Okay, well, thank you all for your time today to listen to our updates on accelerating flowering and maturity in Camellina. And I'll just start with an outline for the presentation. I'll provide a little background on Camellina, our germplasm collection, which was accessed from US and international seed repositories. And in this germplasm, the identification and evaluation of early flowering and maturing accessions, as well as our germplasm development within the University of Minnesota, our mutant population developed by treating Joel seed with ethyl methane sulfonate, and identification and evaluation of early flowering and maturing mutants. And then I'll get into some future objectives for Camellina improvement. So a little background on Camellina. It has two growth habits, a spring and a winter growth habit. The winter lines require cold temperature before they will flower. That's known as vernalization. It's largely self-pollinated. And the seeds are contained in little pods called silicles on the upper half of the shoot. It's extremely winter hardy I've seen it do well on the border of Canada, up in Roseau, Minnesota. It's an allohexaploid with a 785 megabase genome, so bigger than pennycress, which makes it more costly to sequence. But that's a consideration that affects some of our decision making, and we have to be a little more efficient in how we go about it. So advantages of Camellina as an oil seed: it's got a high oil content, 35 to 45% by weight. The oil is low in erucic acid, which is great because it's, I know we haven't obtained GRAS for it is my current understanding, but it makes it more feasible to attain the generally recognized as safe status. And so hopefully we'll be getting that soon. I think, yeah, both Colin and Connie have been working on that. I forget who's, I think Connie's been spearheading it more, but yeah. Yeah, so hopefully that'll be coming soon. That'll be exciting. It's got a relatively low glucosinolate content, also important for GRAS status. It can be used for jet fuel, and the high omega-3 can be considered advantage or a disadvantage depending on how you look at it. It also has high tocopherol content, vitamin E, which can help stabilize that omega-3 so it doesn't become rancid as quickly. But a lot of the kind of commercial food applications still don't find it stable enough, even with the tocopherol. So depending on how you look at it, omega-3 can be great for, you know, healthy oil for kind of some health applications. But for more kind of like the packaged goods, it's not as useful. So primary challenges for growers in industry, I'd say one of the main ones we're facing right now is it's got relatively late maturity, especially compared to pennycress. Pennycress can be harvested in mid-June, whereas Camellina is typically harvested in late June in Minnesota. And this is actually a picture of pennycress here, but the relay cropping idea is the same with Camellina. We plant soybean in early May directly into the standing cover crop. And Russ Gesh and others have shown that the relay cropping method seems to optimize both the oilseed and the soybean yield compared to double cropping, where the soybean is planted after the harvest of the pennycress or Camellina. So since this is the system we're targeting for Minnesota and the upper Midwest, the earlier we can take those oil seeds off, especially in terms of Camellina, the better. Because in my master's, I found that the canopy of the cover crop, whether pennycress or Camellina, but more so, actually more so pennycress, but Camellina as well, creates light stress on the soybean, and the soybean gets etiolated. And this in turn leads to ultimate reduction in yield of about 10 to 15%. So the quicker we can get those oil seeds off, the better. And so that's a primary challenge. Meal quality, again, related to still a little bit residual glucosinolate content and a little bit of fiber. The oil quality I touched on, depending on how you look at it, omega-3 can be good or bad. Seed size is still very small, and yields can be improved a lot, especially with what we're seeing with the spring type lines. We have a long ways to go for our system with the winter type lines. And so to get a little bit more into this spring and winter distinction, the vernalization pathway in Arabidopsis is mapped out here on a gene level. And I'll just briefly go through it, especially on, yeah, this vernalization pathway here. So it starts when cold temperatures influence the gene called defect in VIN induction 1 to start to promote expression of the VIN3 gene. And once this begins expressing, it begins to block FLC, the flowering locus C gene, which is a repressor of the flowering time gene, which is the big player in making flowering start to happen and transitioning from the vegetative stage to the flowering stage. And so it's basically kind of like a cold sensitive timer. And if one of these genes gets knocked out randomly, that is kind of what can lead to a spring type Camelina. That's the distinction there. And as far as the germplasm collection, getting into that side of things, the objective was to collect and characterize as many winter Camelina lines as possible, because that seems to be what will be the best fit for Minnesota. Trying to get those ecosystem services in the fall, and trying to get a good early harvest in the spring, as early as possible. So I screened 438 accessions from germplasm repositories or breeders from Austria, Poland, Canada, Russia, and the US. And here's just a brief kind of synopsis of what I found in a fall planting of this germplasm. It was back in 2015, planted on September 1st. And the vast majority of the lines were spring type lines. And all of the spring type lines did not survive the winter. However, there were some spring type lines that were slow in bolting. And I actually at the time was unsure whether or not it was a spring or a winter type, just because it was hard to phenotype in the fall. It's like, is this winter, is it spring? It's really not bolting as much, kind of as you see in the picture. 226 ended up being a winter line for sure. That was a clear one. 227 was a clear spring line, but there were some kind of in between. So I followed up with a greenhouse test of these slow bolting accessions and also the very slow bolting accessions, and found that there were, of course, a lot of the winter types, but some of those slow bolting ones were spring types. They, without vernalization, without those cold temps, they still eventually did transition to flowering. So that was interesting information, and it led us to determining the number of winter types we had. But first, I'll kind of just provide a general synopsis of the spring types. And the top 5 for yield came from the breeder in Austria, Johann Bollmann. And also, surprisingly to me anyway, I kind of thought that just the raw accessions would not be as high as yielding, but there are actually some quite high yielding accessions just straight from the seed bank. So that was pretty exciting to see. And in terms of earliness, also, yeah, some good ones from the seed bank, straight from the seed bank. So yeah, I think some of these spring lines eventually we can leverage into incorporating some of the markers and genes causative for these phenotypes into our winter lines, ideally. And sorry, I have it paused here, something going on. Trying to get to the winter lines now specifically. Let me try to escape out of it. First, I'm gonna have to stop the share for a second, sorry. Okay, let's see here. I think back on.
Oh, just so you know, what's behind there, Matthew, is all our boxes of seeds. And there's boxes and boxes of Camelina back there that Matthew's collected.
Yes, yeah, we have a lot of mutants, that's for sure. Okay, is it back on? Can everyone see? Or David, you can just let me know.
It's
Yep.
good.
Okay. Let me present. So there are likely just 10 unique winter Camelina accessions. So very limited germplasm in terms of winter types. And so this is another reason that we'll definitely need to incorporate spring types into the mix. So in total, there were close to 20, but a lot of them have a very similar or the same exact name. So that's why I've listed them as a possible duplicate. So very narrow germplasm in terms of the winter Camelina accessions. So in the field in St. Paul this past spring, we analyzed the winter types and some mutants in randomized complete block design with 3 reps. And here is the height data that we collected. A decent range in height. Accession 210 and 259 look very similar. They're actually fairly unique looking compared to all of the rest of the accessions. They have very long, internodes. They have very long internodes compared to the other types. So I wasn't surprised to see that they were taller than most of them too. And Joel is kind of the classic one that we've been using agronomically. And so it's towards the top, but definitely there are other taller ones. And this is relevant because in pennycress, Catherine and others have found that there's a fairly strong correlation, positive correlation between height and yield. And that is, you know, a little of a different story than the dwarf story with wheat, but that's a very special dwarf gene in wheat. So in general, the pattern is the taller, the more yielding. So for maturity in flowering, the range was 15 days for flowering and 12 days for maturity. So the window for maturity kind of was compressed a little bit, but still pretty decent variation. And it's helpful for selecting future parents. And I'll get a little bit more into that in coming slides here. So I made some crosses back in 2017, and my rationale at the time was good by good, or in this case, early by early. And the earlier, the better, I was thinking. And this was not based on any phenotype data from the field. It was just based on what flowered earliest in the growth chamber, because I just planted them right away when I got them in the mail from the seed banks, and just saw in the growth chamber, it was the winter time, saw what did the best. And those are the crosses I made. However, certain combinations of winter lines actually can give rise to more of a spring type, it seems. This is an F5 progeny from accession 399 and 264 from a preliminary yield trial that Catherine planted out on September 14th this year. And the photo I took on December 6th, and it's looking pretty rough. It started bolting pretty heavily, and likely, I would say, won't survive the winter. So this is kind of surprising. Although in some ways not too, I suppose, you know, there is an additive effect. I was kind of thinking that the earliness was, well, I was probably hoping the earliness was going to come from a pathway outside of that FLC vernalization pathway, but it seems in this case, the earliness was coming from that vernalization pathway. And so combining maybe weaker alleles from 2 different parents led to even weaker vernalization and bolting in the fall. And we saw fall bolting for 1 parent in the past, in 2016, and also for the other parent this year in 2020. And this fall, I mean, it was mild, but it wasn't unusual. I went back and I looked at the past 10 years of weather data in St. Paul, and 4 of the last 10 falls have been equally warm or warmer on the St. Paul campus. So this is something that is gonna be an issue for us moving forward for sure. So it's gonna inform our crosses, it's gonna inform the parents we pick. And this data is fall bolting notes that Catherine took from both St. Paul and Rosemount this fall. And I'll orient you to the table here. So on the top row, I've got the labels, so the name of the accession or the cross, the frequency at which the pedigree shows up in a certain group, whether it was bolting or not bolting. And then of course the pedigree, and then St. Paul is in pink and Rosemount is in blue. And so both St. Paul and Rosemount had a number of the 399 by 264 pedigree crosses bolting. So those likely won't survive the winter. There was a little more bolting in St. Paul than in Rosemount. And there were still a decent number of F5s that had not begun bolting, but I would expect those also to not flower earlier than their parents. I mean, we'll see. I'll find out this spring, but I am seeing that a pattern of an additive effect in the FLC pathway, in the vernalization pathway. So if we're not combining those early traits in that pathway, we're probably not going to see any earlier flowering than the parents. So it's a little more complex and in some ways disappointing, but also it's motivation for finding new ways to tackle this earliness problem in Camellina. So the F4 progeny evaluated in the spring of 2020, some of them, this 1378-1 was just like before the 264 by 399 parent. So just like the ones that we saw bolting this fall, in fall 2019, they didn't bolt and they did pretty well. It was earlier than the parent, than either parent, so that was a good sign. However, a spring cold snap got it, unfortunately. So that's another thing we have to deal with on the spring end. I looked at the past 10 years of weather data on the St. Paul campus for April low temps as well. And it was a 2 in 10 cold snap, 2 in 10 year cold snap that happened. So again, it's not like a 1 in 10, it's still somewhat common. So something we definitely have to think about. And so planting dates, coming back to planting date, it seems like that's going to be very important for us moving forward. And Rush Gesh and his colleague Steve Cermak found that Joel planted in late September to mid-October produced the greatest seed yields. So that is something that we're definitely going to have to bear in mind more so moving forward for both these early flowering accessions, and I'll be getting into some mutants here in coming slides as well. And for the mutant population development, it was based on the variety Joel, which Russ was able to get from his collaborators. And it originally came from TCI, which, well, I guess it was King Seed, but it was overtaken by TCI, Technology Crops International, in collaboration with researchers at the USDA in Peoria, Illinois. And so Rush has been using this genotype for years now, and this is what we based our mutant population on. And we use ethyl methane sulfonate. And this is kind of, this cartoon up here is showing what chemically goes on with ethyl methane sulfonate. So it transfers an ethyl group to the guanine, to the oxygen in guanine. And this forms 6-ethylguanine, and it will no longer bind to cytosine. Instead binds to thymine. And so in this case, guanine is acting more like an adenine. And this is why the majority of EMS mutations are C to T mutations. And so they're single base pair mutations, and this can lead to kind of weak alleles, like missense mutations, or stop codons, like nonsense mutations. So very strong alleles or even weak alleles, and both can be useful in terms of shattering, like we've shown in pennycress, that a weaker allele, it's like we don't want it to be too shatterproof, because then you can't really even break it open with machinery very easily. But you want some level of shatter resistance. So EMS is a great tool for that. And we planted out the mutant plants in the field, and each row contains M2 plants, and we harvested those individual plants and planted them out in large populations of 3,000 in St. Paul and about 1,500 in Moritz. And we're able to keep the population advancing. And in 2018 found, or 2019, excuse me, found spring 2019, found good variation in our Camellina mutant population. There's some wrinkled leaf, waxy, but probably most relevant for agronomic purposes would be the tillering, especially the branchy, I would say, just based on a presentation that I saw in the Agronomy Society meeting this fall. Ramdeo C. Paul presented on Carinata, which is another oil seed, and found that greater branching in genotypes of Carinata led to higher seed yield. So that might be interesting agronomically to look into some more of those Camellina mutants as well. And then of course, the early flowering maturing mutants are very relevant for the relay cropping system. So in Rosemount, in May of 2018, I identified a mutant that was flowering before anything else in the entire field. So I was pretty excited that day when I found it. And I flagged it so I could come back to it, and told everyone about it. And this is what it looks like compared to Joelle. And we saw in subsequent years that it was a heritable trait. So that was exciting to see. And just consistently performed well, as you know, planted in various planting dates in the fall even. Another mutant that we found in May of 2019 was, it was just numbered 336-1. Since we found it in St. Paul, we're calling it STP 336. And kind of similar, it was a little bit later than Rosemount 9. I'll show some of these data here. So in this 2020 evaluation, which was randomized complete block design, 3 reps, compared to pennycress, Joelle, the wild type, is later in both terms of flowering and maturity. But with these mutants, these Camellina mutants, we're seeing the maturity and flowering kind of approach pennycress, or actually in terms of flowering even get faster than pennycress. So yeah, this was exciting to see. Hopefully, you know, we can leverage maybe some of these mutants to get to a harvest of Camellina that's on par with pennycress. And in terms of height, they are a little bit shorter mutants, but maybe with some back crossing, which I have made some crosses with these, we can get the heights back up and still hold on to the earliness traits. And these mutants also have been planted in a multi-location yield trial in collaboration with Russ Gesh and Morris and Catherine Frells in Rosemount. And so it'll be exciting to see how they do this year. I know from what I hear, they're looking good so far this fall. So yeah, hopefully we'll get some interesting data this spring. I also did a planting date study for these early maturing lines. It was a randomized complete block design with 4 reps per planting date. The first planting date was early September, and the second one was in mid-September. And you can see the map and the mutants and accessions we used. And I'll just kind of jump into what we've been finding here in the fall. And we're actually getting some bolting going on with the earliest planting dates of especially the Rosemount early flowering 9 mutant, and a little bit in the St. Paul early flowering 336 mutant. You can see some little red tags in the photos. I've tagged plants that have been bolting just to see if they end up surviving the winter, as we have seen them survive, or at least we haven't seen bolting plants necessarily survive, but we've seen the mutants survive in general in the past 3 years. So it'll be interesting to track that and see what happens. But for the second planting date, oh wait, I'll get the second planting date next. But just quickly wanted to show a little bit of notes I took that does seem to be more of an issue with Rosemount 9 than St. Paul EF 336. But for the second planting date, we're not seeing any fall bolting. So planting date for the mutants seems like it's going to be very important as well. And these lines were also included in Catherine's preliminary yield trial. And that was planted on September 14th. And there's no evidence of bolting there either. And I'm showing that picture of that 399 by 264 F5 again, just for comparison as well. So that's kind of an encouraging sign that it's not as bad as it could be, like with some of these other genotypes that we have. But it's still maybe a cause of a little concern. Although fall bolting has been a problem for early pennycrest lines as well. I've looked at some early flowering pennycrest lines. This line A725 started bolting in mid-October, and it was planted in mid-August, so it was on the early end. But as we try to push flowering and maturity earlier for both pennycrest and Camellina, we can kind of run into an issue on the other end with planting. So it could be an issue for both crops moving forward. So I've made crosses between early flowering accessions and these early flowering and maturing mutants. And I'll get into those here in coming slides. I saw segregation, and I've got the segregation ratios and kind of what we expect. So it appears to be a 2-gene situation, kind of the 1 to 15 ratio, the classic 1 to 15, 2 recessive genes. And my initial thought was that 1 recessive allele is coming from the accession parent, and the other is coming from the mutant parent. And that's why we're seeing the ratios that we do. But to be determined. And a little further analysis of these lines just planted very recently. It's a vernalization requirement experiment, and it's a randomized complete block in the grow chamber with 7 reps, planting various treatments of days in the cold, and going to start those here pretty soon. But just this morning, I checked on this experiment and found that the Rosemount EF9 and the St. Paul EF336 are beginning to bolt without any vernalization. And it's similar to this accession 221, which is one that I had noted back in 2015 as being slow to bolt as a spring. So it appears that they're intermediate spring types, and the F4 of this accession 226 by Rosemount 9 is an intermediate spring. However, to my surprise, the F4 of 226 by St. Paul EF336 is winter. I was able to harvest the seed in time for planting this fall for the first cross, but not in time for the second cross. So I'm field testing the first one. I've got a picture of it here, and it was planted on September 23rd. And I flagged plants that came up just to verify that indeed, yeah, this came up in the fall, and I can evaluate it again in the spring. It was a size of 49 population. And I also had checks in there, so it'll be interesting to just evaluate the checks again too, for another planting date. The parents and Joel as well. So yeah, it'll be interesting to see what happens with this planting date and if we get additive effects in the accession by the mutant cross here. So yeah, spring is shaping up to be an interesting season for note taking. So just brainstorming some ideas of, you know, if falls that are mild are going to be a problem for us, maybe there's something else we can do more from like an agronomic side of things. And I did a little research and found that an ethicon application can kind of suppress flowering a little bit. And you can see in this photo here in the species Angelonia that you get a little bit of suppression at 100 parts per million and a lot of suppression at 200 parts per million. And they did this as a foliar spray, but I'm curious if maybe a seed treatment may yield similar results as well. But just a little brainstorm, throwing it out there, may be an interesting area of future research, may or may not ever be applicable. But and on the other side of things too, some plant growth regulator applications in the spring may help. Gibberellic acid has been shown to speed up flowering as a foliar spray. And then I just randomly stumbled across this carbon nanoparticle can help speed up flowering in Arabidopsis. So that's, you know, another way to tackle it perhaps. And then also from a reverse genetics approach, there are Arabidopsis mutants in the gibberellic acid pathway. I'm just showing one down here. We probably wouldn't pick this one because it is involved in disease resistance as well. But there are other ones in this pathway that may help on a different pathway outside of flowering locust C. So other approaches may be interesting. So other future directions, planning crosses between the winter and the spring types, even using that population I'm screening for the vernalization requirement. And based on some research done on flowering locust C expression in Joel and a spring type, a known spring type, Colorado 46, we're seeing high expression of one of the subgenomes of FLC and CO46 but it's been shown to have a frame shift mutation in FLC. So even though you're seeing expression in one of the subgenomes for FLC, it's likely non-functional. So basically, nothing going on in CO46 for FLC in function. But for Joel, two of the three subgenomes have a functional FLC. So we would expect 1 in 16 F2s of a spring by winter cross to have 4 functional FLC alleles and 2 FLC orthologs. But kind of the ecological idea, if they don't use it, they lose it, reference to spring types for the whole FLC pathway, it's probably not just FLC that's non-functional in the spring types. There's probably some others in that pathway. We just have no idea how badly that timer is broken, basically, or how many parts in that timer are broken. So it could be a situation where we're going to have to do a lot of back crossing to get the best of what we want from the spring types and incorporate them into the winter types. And here's just another example of that in this photo. David made a cross, I think way back in 2015, of CO46 by Joel. And even the F1 had an intermediate type, intermediate growth habit, as you can see here, which is kind of what we would expect based on this data. It seems to be that a heterozygote is not enough, not strong enough FLC to suppress flowering all the way in Camelina. That's a different story in pennycress. A heterozygote FLC is strong enough to suppress flowering, but not the case in Camelina. And so this is probably a clue as to why we're seeing this kind of intermediate spring type in Camelina. So this was kind of really focused on flowering and maturity today. But we also have future targets that we'll be working on as a lab. And Ratan, I'll be collaborating with Ratan. Ratan is really leading a lot of the work on the oil and protein and looking at those, at all of our accessions and mutants with NIR. And Ratan's done a lot of the work on the genomic resources as well for the Camelina. And Christian will be continuing some of his work on gene editing to improve Camelina meal quality. And I also know that Catherine is in talks with some collaborators for Camelina as well. So we've got some future directions, and yeah, just hoping to keep improving Camelina and get it to where it needs to be. So with that, I'd like to thank the team and my advisor, David, for his guidance along the way, my committee, really helpful and giving me insights and guidance, and just, yeah, a lot of help from all of the postdocs, Ratan, Catherine, Christian, and Kevin. I couldn't have done it without the technicians, all listed here. A lot of undergrad help for the Camelina project. Really appreciate all of Russ's and Frank's and Jim's help over the years. And the Forever Green Initiative for the funding of the research, as well as all of the other funding that we've gotten to support this work as well. And I can open it up for questions and discussion. So thank you for your time and interest.
So any comments or questions for Matthew and the rest of the team? You can put it in the chat. I think we're all set up to open it up. I hope if you raise your hand to ask a question directly.
Matthew, there is a question in chat from Carmen. Have you seen any correlation between bolting with the daylight or growing degree units?
Yes, that is a good question. And I just made a very kind of non-experimental observation just recently on daylight. It seemed that a longer day length did really seem to push our Rosemount Elf 9 faster in the growth chamber. Yeah, that is something, that's a great question, and something that we'll have to do some more investigating on in some controlled light experiments. I think that's a great future direction as well.
There's another follow-up question from Jessica about how, what do you think, is actually earliness a breeding target, or it's more of a management issue?
Oh, okay, sure. Well, ideally what we want is earliness in the spring, but not in the fall. We want early bolting in the spring, just so that we can get the crop off of the field, open up the canopy for soybean to reduce that light stress on the soybean. So it's kind of going to be both a management issue and a breeding target as well, to kind of walk that fine line and really try to get the best of both.
There's another question from Gary Gardner. What is the critical day length for flowering in Camelina?
So this I will have to do some more reading on, actually. Yeah, that's a good question. And yeah, I've got a few holes in my knowledge I have to fill in. So I'm sorry I can't answer that, Gary. But thanks for bringing it up, because it does seem to be important. And in terms of when these plants are actually, you know, flowering in the spring, it's in April or early May, late April or early May. So it's, you know, kind of a longer day situation, but definitely something that will require some more research.
Okay, one more question. So what are the primary barrier for Camelina seeds to be used as a food and feed? There's not like barrier necessary. I can answer some of it.
Sure,
like,
yeah.
Camelina can be used both for food and feed purposes. I mean, at the moment, like it's the oil is being extracted, and you know, you can buy it in stores. In terms of feed, there are some studies going on. The protein quality is good. That's what Pam Ismail is working on, trying to understand how Camelina protein can be useful as a plant protein for different products. And her student Vaide has been looking at 4 lines that show some variation in certain like certain protein bands, but the characterization is in progress. Maybe she will have an update in the coming spring. Okay, Matthew, there is another question.
Yes. Yeah, I'm looking at the chat now here too. So let me.
It's from Corey.
From Corey. Okay. Okay. On the article that showed the correlation of gibberellic acid, are you saying that planted early with the plant growth regulator contains this would help prevent early fall bolting? So yes, Corey. So actually, let me go to that slide again really quick so I can touch on that. Well, let's see here. Sorry, having some issues. Basically, there are two plant growth regulators I was trying to highlight. One is ethifon, and I was thinking of ethifon, which is in the ethylene pathway. I was thinking of ethifon more for a fall application. And that would be for genotypes that are at risk of the fall bolting, to kind of suppress that. But on the other end, so yeah, there's the ethifon. So I was thinking of that for a fall treatment. But for spring, I was thinking more of gibberellic acid in the spring on lines that have a strong flowering locus C allele, or that can be later maturing. So that you can kind of speed up maybe these later lines in the spring. So you can kind of maybe tackle it in the fall genetically, in a sense, or genetically early line, and you kind of have to suppress it with a regulator in the fall, or genetically late line in the spring, and you have to promote flowering with the regulator. So yeah, hopefully if you have any more questions, let me know. I'm gonna go back to the chat. Okay, so okay, it looks like, okay, that clarified. Yeah, it looks like everything from the chat. Thank you, Colin, for pitching in on the details for grass. Any other questions?
Yeah, I was wondering if Mark is still on the line. If they were growing some Camellina down there, or some of the winter Camellina, I was wondering if Mark would be able to say anything about that. Maybe not.
Yeah, it looks like he's not on the line anymore.
Okay. That's one of the things that's going on right now, is that we're collaborating, or beginning to collaborate with folks down in Illinois, cover cross and such, to see what winter Camelina can do down there. It'll be interesting because they're going to have a bigger challenge with the warm and cold snaps that they repeatedly get down there. And I'm just curious to see how that's going to play out.
I have a question about the observation. You made a cross between winter tab early by early. You observe spring tab. Does that mean the maturity gene, they are not, they have additive and epistasis at the same time. If they do have epistasis, how will you develop a marker for developing winter type early maturity Camellina?
So yeah, that's a great question. Yeah, I guess I'll have to do a little more thinking and looking at the data, and maybe doing some more experiments to really get at if it is more additive or if it is epistatic. I kind of think at this point it could be either, but maybe I'm wrong. Is there something that gave you a tip that you saw in my data that made you think it was epistatic?
Well, because you make a cross between winter type early by early, we expect early.
Right.
And we don't expect any spring type, but you do notice some of those.
Yeah, I guess,
spring type.
right. So I guess what I'm thinking on that is that probably, at least my hypothesis of what's going on, I could be wrong, but so one of the early winter types has, I mean, it's a hexaploid again. So it has kind of a weak allele or a series of alleles for one gene in the FLC pathway, and another of the winter types has a weak allele in a different gene for the FLC pathway. And then when you combine those, you're kind of seeing an additive effect. Okay. So that's my hypothesis.
And as a breeder, and will you consider make a cross between winter tab early by late? Then you can further confirm whether you can see even early or even later for the additive testing.
Yeah, that is a very good idea and point. Yeah, so I have that late flowering and maturing spring type and the early spring type as well. So I think making crosses of all the, whether it's spring or winter, just making all the pairwise crosses for whether it's late or early would be great. Yeah, just for marker identification and for developing, resources and trying to develop the best lines. But I will definitely make note of your point that it could be epistatic. I'll try to pay attention to that to see if we're getting something that is kind of more than the sum of its parts.
Okay.
In terms of,
excellent work. Thank you.
Yeah, thank you for your points and question. Appreciate it.
Are there any other comments or questions? Raise your hand.
I can make a comment. Just simply, I mean, what we do over here in this lab is we find some of these foundational traits, like Matthew's looking for these early flowering things and such. And the thing I wanted to emphasize is that this is just kind of scratching the surface of what can be done in terms of making improvements. We have a lot of different genes we can look at. But at the end of the day, what we're going to produce is something that we're going to be handing over to the breeders. And over time, they're going to be the ones that make the improvements and make this into a commercial, I think, viable crop. And so I just want to make sure that folks understand that I really appreciate and understand that that part of the game. That's it.
Well, as we all know, thanks for that comment, David, because that's really the game, right? We have in the enterprise development around all of these forever green crops, and that's what makes what I think the Forever Green initiative and beyond unique, is that we already have these relationships between the basic work and the breeders and the folks doing the agronomic work, and tied then to the end users. So I think that is really the exciting part of all of this work. Any other comments or questions? And what I always do is remind everyone, we have these lab meetings every week, but also, obviously, I would love, and David and others would also encourage everyone to continue to follow up and provide feedback to these team members. And the reason for these meetings, these are not seminars. These are designed to be presentations for discussion and making sure that there's follow up on all the ideas that are presented in this conversation. So